Alright, hello and welcome back to these little physiology talk-throughs in video. So my name is Dr. David Green and I'm going to talk to you now about the main concepts that we see in physiology for the endocrine system. Now, once you get the understanding of what hormones are, that they're chemical messengers released by endocrine glands. Endocrine means ductless, so they dump into the bloodstream. So these chemical messengers are dumped into the bloodstream. Because the bloodstream reaches every cell of the human body, then they're basically ubiquitous, or they travel everywhere. Now, these hormones or chemical messengers therefore have access to potentially all the cells of the body, but only the cells that actually have specific receptors for them will respond to them. There are two main ways of stimulating the target cell. And there's one with there's a third way which is very close to the steroid hormone way, and we'll talk about that as a, essentially I think compare and contrast mode. So we have our steroid hormone, and then again we have our protein based or polar hormone. Now I think the easiest way in physiology to separate the hormones is by their polarity, because their polarity dictates how they actually target the cell that they're going to make a change in. So for a polar hormone, which is based off amino acids or polypeptides or proteins, this means that they are water soluble because they have polar charge on their molecules, and that they can't enter the target cell because proteins in particular are simply too big to get through the phospholipid bilayer of the target cell. This then means that they have to create something called a signal transduction, which creates a secondary messenger inside the target cell. Now that's what I'm going to look at in the next video in the sequence, so we'll leave that there. For this one, we're going to look at the non-polar hormones, which are the majority of which are steroid hormones. Now you may recall from about week 3 we looked at macromolecules and we looked at the lipids, and we said that the group of the lipids are contained of the fats, which are the triglycerides, which we consume in our diet, and it's the stuff that I like to collect around my abdomen for some reason. And the other group in the lipids was the steroids, which are these circular or cyclical structured molecules, with the base molecule or starting point being cholesterol, which again we consume in our diet and we can also make in our liver. Now the steroid hormones are predominantly non-polar, meaning that they don't carry an overall charge, therefore they do not interact with water, which makes it difficult to get them through the blood by themselves, so they require a protein transporter, in most cases an alpha or beta globulin. But because they are non-polar in nature, they can actually pass through a phospholipid bilayer quite comfortably and access the cell. Not only that, they can also cross the phosphobilipid layer, protecting the nucleus of the cell. So a non-polar hormone can not just get into the cell, it can get all the way to the nucleus and affect the DNA directly, and that's what this talk's going to be about in terms of how they do this. So behind me on the board I've written most of the stuff down. You'll see similar diagrams in your notes and your PowerPoints, so we're trying to keep it as similar as possible to make understanding as straightforward as possible. So I'm going to turn around now and let's talk through the process. Alright, so we're looking at a steroid hormone here, and the steroid hormones are non-polar, so they are, and I should probably put that up there. So steroid hormones are non-polar, they carry no charge. So they transfer down through the bloodstream. I've got the hormone in green here, and then the blue part is a protein transporter. So they pass down through the blood, they are able to come out through the capillaries, and as they do, they make their way through the interstitial fluid. I can't stress enough how important it is to remember this space between blood and cell is the interstitial fluid. It's the 66% of the extracellular fluid in the human body, the rest being found in the plasma of the blood. It has to pass through there and make it to the outside of the target cell, at which point the hormone is dropped off and it can pass through the phospholipid bilayer of the cell. So let's put that in, it's the cell membrane. So the hormone has now made it inside the target cell. What happens next? Well, it's picked up again on a transporter and carried over toward the nuclear membrane. It's dropped off and it can move across the phosphobilipid layer of the nuclear membrane because again it's still non-polar. When it arrives inside the nucleus, we have structures like this called HREs. We've got one up here, hormone receptor elements. And these are basically two molecules joined together. The hormone is picked up and joined in here. So that is the binding site for the hormone. And then this part here is a binding site for DNA. And that's important because then the target gene, the DNA, is opened up and this hormone receptor complex, because it's now got the hormone bound to its hormone receptor site, can now bind onto a half site here. Now these are half sites, so it takes two to bind in order to stimulate protein synthesis of the target gene. So, to recap, non-polar steroid hormone transported in the blood, attached to protein transporters because it's non-polar and doesn't dissolve in water. These are released at the capillary beds and they make it out toward the cell membrane of the target cell. Hormone is released, passes across the membrane, picked up by another transporter, taken to the nuclear membrane, released and travels across the membrane again, where it's picked up by a specialist molecule called the hormone receptor element. It binds on the hormone binding site and that activates it and allows it to find a half site in front of a gene and it binds there. If there are two and they both bind either of the half sites, this causes expression of the target gene, protein synthesis then ensues, transcription comes out, translated into a new protein. And that is how a non-polar steroid hormone stimulates its target cell. So in essence, a steroid hormone creates the production of new proteins, and in many cases these new proteins are enzymes which will catalyze new reactions. Now, there is one other type of non-polar hormone that we need to consider, which do not fall into the category of steroid hormones. These hormones are released by the thyroid and is known as T4 or tetroiodothyronine. It basically does the same thing as we've got here, but there are two differences in a T4 molecule stimulating a new protein and a steroid hormone stimulating a protein. And their classic questions are compare and contrast the action of a steroid hormone and a T4 hormone on the target cell. So you would basically explain this for most of your marks, and then to get the contrasting action of the T4, you have to point out where the two differences are, and I'm going to show you that now. Alright. So we've got the T4 hormone. So let's just put the T4 hormone here. Let's give it a new protein transporter. Oops. Using the wrong colour. Keep getting your colours right. Alright, so here's our T4. Just the same way. T4 makes its way out to here, to the cell membrane, and then the T4 comes inside the cell. So far, T4 and the steroid hormone have done exactly the same thing. They've used a protein transporter to get through the blood, be released into the interstitial fluid, arrives at the outside of the cell membrane. The T4 and the steroid hormone are released. They're both nonpolar, so they can both cross the cell membrane. So here's the first difference. 1. Now you might find that a little bit difficult to read, but T4 is cleaved or cut into T3. Now what do the T's stand for? The number of iodine molecules in the hormone gives it T4 or T3. Now the active version of thyroxine, which is the hormone released by the thyroid, is T3, so 3 iodine molecules, but it travels in an inactive state in the blood predominantly as T4. When it arrives inside the target cell, it gets cleaved, or one of the iodine molecules is released, and it's converted in the cytoplasm to T3. So that's the first difference. T4 is activated into T3 inside the target cell cytoplasm. So now we have T3 being transferred over here, and then T3 crosses over and enters the nuclear membrane. So once again, T3 will find an HRE that binds it, like that, and that will bind a half site. So again, we'll have so there's our half sites. So now we've got our hormone receptor complex with T3 bound on one of the half sites. Alright, well here comes our second change. There was our first change, T4 to T3. Our second change is what binds to the second hormone receptor element, and it's not T3. So on this one, we have a derivative of vitamin A called 9-cis-retinoic acid, and it binds to this half site, and T3 with its hormone receptor complex binds to the other half site, and that causes protein synthesis to take place. So, recap. Steroid hormones and T4 are non-polar. Therefore, they require help to get through water environments, so they have to be bound to a protein. But they can pass cell membranes of all types because they are lipid-based, and so they'll just float through them. So, steroid hormone travels through capillary bed just the same way as T4 does, bound to a protein, comes out of the capillary, arrives outside the cell membrane. Both T4 and the steroid hormone are released, and they're able to move into the target cell straight through the phospholipid bilayer. They are both then picked up by a protein and carried over to the nuclear membrane. But here's the first contrast. T4 is cleaved to T3 at this stage as it enters the cell. So T3 and the steroid hormone now arriving here at the nuclear membrane, again both released, they both diffuse across the cell, the nuclear membrane, and then they're both picked up by the hormone receptor elements, making a complex. In the case of the steroid hormone, 2 steroid hormones bound to their hormone receptor elements on the half sites causes protein synthesis, but the second difference between steroid hormones and T4 hormone is that only one T3 is required on the half site, and the other one requires a derivative of vitamin A called retinoic acid. Now if you're writing this in an essay, I mean, as long as you remember it's one T3 and the other one is another chemical, getting a derivative of vitamin A will get you the points. Obviously retinoic acid also the points. But this is how non-polar hormones stimulate the target cell. They do so at the DNA level, and that is how they do them. So thank you for listening.